recorded. If you would like to ask a question to the speaker, please use the Q&A function at any time, and I will ask your question at the end of the talk. If you need to contact us about anything else, for instance, technical issues, please use the chat function and one of our team will get back to you. The recording will be made available for attendees after the event via the email that you used to register for the lecture. As regular attendees of these lectures will know, this year's theme has been paleobotany and the evolution of land plants. Most of the previous lectures focused on the earliest land plants, so it's fitting that our final lecture will look at the youngest of land plants, the flowering plants, the origin of which Darwin described as an abominable mystery. It is therefore my great pleasure to introduce our final speaker, Dr. Julia Gravendyck from the University of Bonn, where she is a lecturer in systematic botany in the Bonn Institute of Organismic Biology. And it's great that our twin city of Bonn is represented this evening ahead of us reinstating our memorandum of understanding with the University of Bonn Botanic Garden. So without further ado, Julia is a palynologist, which is someone who studies pollen, and she has a particular interest in the pollen of the Mesozoic. That's the period from the Triassic to the Cretaceous. And the insights pollen found in the deposits from those eras can give us about the plants that were around then, their evolution, and the vegetation of that period. To do this, she uses a range of microscopy techniques to study fossil pollen, principally light microscopy, scanning electron microscopy, and fluorescence microscopy. Julia studied for her PhD in the Division of Botany at Freie Universität Berlin, where she used pollen to investigate the vegetation dynamics of the Triassic-Jurassic transition. From there, she moved to Hanover's Leibniz Universität to work as a postdoc on a project aimed at deciphering the very early phase of angiosperm evolution using palynology. Since 2022, Julia has been at the University of Bonn, where her palynological research, getting my words twisted there, asks, can pollen indicate environmental stress? And where did the very first flowering plants appear? Julia, please tell us the answers in your talk if you can. Thank you.
Thank you so much, Simon, for the introduction. I'm going to share my screen. And I try to use this tongue twister, palynology. We're going to hear a lot about plant evolution through the lens of microfossils. And we talk about microfossils when we talk about pollen and spores. And as you see in this image already, you see pollen and spores as a reflection of the environment. They are not the direct indicator of what we see, but they're sort of evidence. And with this evidence, I try to reconstruct what the past might have looked like. And I'm first going to tell you a little bit about the superpowers of pollen and spores, and then how I'm going to use them to apply those big questions that Simon has just raised. And then we're going to apply this to a little case study to close the session off to talk about angiosperms and the rise of them, which is indeed an abominable mystery. While waiting, you were all primed with this little question, and you might have been pondering, okay, this is about plants, so probably the answer is C. And I hope you're going to forgive me that the terminology is not perfect. Silk of a spider, tongue of a snail, or the sperm of a plant. Well, all of these are actually the top three of the hardest organic materials on Earth. So no matter which of the two you thought it might be, you were pretty much right, because all of them are studied for material sciences because they're all hard or sturdy in their own way. And which is the top one? Well, actually, it is the tongue of the snail, a radula, the tongue that is used to graze off algae off stones. And this is, in fact, the hardest organic material, but only on place two is this. A lot of people might know this from allergy season. This is the pollen of a grass, and you see it is actually everywhere in great abundances. Every little white dot is a pollen. And you might also know it from your beautiful bouquet of lilies that stands on the table and makes this annoying yellow dust on the tablecloth, possibly. This is the pollen, and this is what we're going to look at for the rest of the day. Number 2 in the ranking of the hardest organic materials on Earth. What is it used for? Well, you see here the anther that produces this beautiful yellow pollen that sits on the surface, and ideally, from this male part here around, the pollen gets deposited on the stigma, the top surface that you see here, and from there, the pollen has to grow a little tube and transports the sperm of the plant all the way down here to go down, and the sperm has to fertilize the egg to produce the embryo and the seeds that we all know. So the pollen is an essential unit in this life cycle of plants. If we don't just look at the full presentation of the pollen as we see it here, but look at an enclosed bud as we see it here and cut it, we see this. And here you can see where the pollen is actually produced in these beautiful little anthers. They look a little bit like butterflies, and every wing has two yellow bags. They are the pollen sacs, and every anther has 4, or these ones do. And on the red slit, that's where they break open to present the pollen as you just saw it. Now, what does the pollen look like? It's actually so small, microfossils, that we need a very powerful microscope. And here we are using scanning electron microscopes. So we're looking with electrons to image the beautiful surface of the pollen. And actually the first time that I saw pollen in my undergrad studies, that was really the day that I fell in love with pollen, and it has stuck with me ever since. The shapes are enormous and super beautiful. And it is that surface that you see here that is another power skill of pollen. It is this hardest organic material. And this actually is called spore pollenin. Spores and pollen possess it. And it is very chemically inert. So it's very sturdy, no matter what kind of chemicals you treat it with. And it is very stable over time, especially under anoxic conditions. And that's the superpower. These things can last over millions and millions of years. Actually, the very earliest land plant fossils are consisting of spores because nothing else was there yet and nothing else preserved just as well. So this is definitely a superpower of the sporopollenin, and that is the coat around the pollen, but also around spores. And I call it the mithril of botany because it is protecting the insides, the reproductive unit of all the land plants that are possessing this. Why do all land plants have it? Well, life wasn't always on land. When it came out of water, there were new challenges on land. Light, radiation, UV radiation especially, desiccation, quite dry, you don't have water availability all the time. So how do you protect this inside, which is actually necessary for reproduction? Well, you enclose it into this sporopollenin skeleton, and it also has beautiful shapes, and they can have functions they don't always have. Sometimes I just wonder why there is so much diversity in them. And you can definitely tell, for example, a spore apart from a pollen, but even within them, plants have so many different forms of them that very often you can tell at least a plant group that produces a certain spore or pollen. This superpower is number 1, the sturdiness. The superpower number 2, compared to all other fossils when you study paleontology, is this. it occurs in huge abundances. That is exactly what annoys so many people when they're allergic to grass pollen, especially, but also to many other plants. It is everywhere. And because it is everywhere, we have a lot of it to study in the fossil record. Superpower number 2. You see here that after a pollen rain that you sometimes have during certain times of the year, when especially the pine pollen that you saw in the video before is released, all of a sudden, all at once, and it's everywhere. And just like it is here on the street deposited, it also gets deposited into rocks. And there it can stay and last over millions of years because sporopollen is so sturdy. How do we use this now to study palynology? The tongue twister. Basically, palynology is the study of dust. It comes from the Greek palynos, which means strew or sprinkle. My former flatmate used to call it dirt on glass that you're looking at. Basically, it's just little dirt, little plant remains, and therefore microfossils. There's other fossils that can be studied, which you have heard about in the previous lectures that if you attended them, and especially macrofossils are the things that you see with your naked eye. No additional power needed unless you want to see the intricate details of them. But also mesofossils, meso meaning between the big and what's coming up the small, is, yeah, you need a magnifying glass, but you don't need a microscope yet. So these are the other fossils that people are studying, but that's not what I do. I always like microscopy, so I picked something where I always need a microscope to actually visualize what I want to study. So now you know 2 main superpowers, sturdiness and abundance. How can we put this to use to study plant evolution in the fossil record? You can actually do all kinds of things with pollen. You don't have to go into the past. You can also do extant studies. And for example, here you see pollen from my class of this winter semester. And these are beautiful microscope images, SEM microscope images of the pollen that we found in trap nests from wild bees. And we're using them to match the wild bees with the plants that they're feeding on. And sometimes some of the larvae that you find in here are not even classifiable without the pollen source. What we're doing here is creating a network of plants and animals, and we need the pollen to do so. You can do this today, but you can also do that in the past. This is a cool paper that came out just 2 months ago from the working group in Vienna, and they actually specialize on not so old material. So we have a lot of better preservation because it's not so old. Here you see a flower. It actually still looks like a flower, although it's just an impression. And here you see a bumblebee. And now, what can you look at to match those two together? Why are they shown together? Well, you would have to now use the microscope and look at the pollen that you still find in the flower and the pollen that is stuck on the hairs of the bumblebee. And what they found was that there's actually the same pollen. And what they are using to do these plant-animal interactions is to match flowers with insects in the past. And you can do that using the pollen that match on both. Pretty cool thing. If we want to go even deeper, we usually call it palynology. So the deep time palynology. And as Simon mentioned already, I'm mostly interested into the Mesozoic, the Earth Middle Ages, if you want. And in these older times, the amount of fossils that we have, especially macrofossils, are severely diminished very often because it's quite old, things break, they're not preserved as well. And in general, fossilization is an event of luck anyway. So the deeper you go, chances you find something nice and workable is even rarer. So working on the small stuff might be more informative. And sometimes you go to a museum and you see beautiful reconstructions like this, and you might wonder, how do they even know that it looked like this? Well, option number 1 is you find nice macrofossils, remains of the plants that you see here, for example, these horsetails in the back, or these araucarian trees in the foreground. Well, if you're lucky, that works, but very often you're not. And then often what paleontologists have is nice bones of dinosaurs, which they want to show, but they don't know what plants surrounded them. So then they come to a palynologist and ask them, so what do we make in the background, the backdrop of the vegetation, because that is the foundation of all that life. And we usually take a little bit of rock of the remains, the bones very often, and then we study the pollen and the spores that are enclosed in that rock, and we match what could that be, and thereby reconstruct this environment that you see here. And this is what it looks like. Beautiful, manifold shapes. And I am so surprised that nature still finds new ways. There are millions and millions of years, and so many things that all look different. It's incredible diversity. And through these microfossils, these pollen and the spores that you see here, we can actually tell the environment. And when you look at these spores in detail, you see the intricate details, and they all differ a little bit, but you need to look in detail because otherwise you might miss them. The size of these spores and pollen, if you imagine you would be a bee and holding a pollen, it's pretty much like we standing and holding a soccer ball with us. So that's about the size range that you see here. And some of the things are so typical for certain plant groups that we can match them to the parent plant. Some of the things like this thing here, if anyone knows what this is, I would be so happy. No one really knows. This is a big enigma, like many of those old things that we can't really match. But you see another aspect here. You see that it fluoresces here on the right. And those of you who attended the lecture by Jenny McElwain last time, they have heard about fluorescence already. Fluorescence works very similar to the phosphorescence, which you might know from the glowing stars you might have had in your child bedroom. Basically, you shoot at something with light, and the molecules interact with this light, and you can detect what is coming back in a certain way to image this thing here. And this is possible because the thing that interacts, the molecule, that's the sporopollenin. And the sporopollenin here is still the original. Just like the cuticle in Jenny McElwain's talk, the sporopollenin is still the original material. It's millions and millions of years old, and still it's preserved. The content of the cell is actually gone, but it's not replaced by something else. It is still the original. And that's the thing that makes it glow here with the fluorescence. Now, if I find an isolated spore, how do I actually know what parent plant produced it? Like in the former case where I said I have absolutely no clue what this might be. Well, ideally, at some point in time, one of the researchers who works on macrofossils, the big things, can find a pollen or spore producing unit. And then you look at the pollen and the spores that are inside, and thereby you can match these things with the parent plant. Ideal world. And for many things, we can do this. For everything where we don't have these features, you might actually have learned what certain characteristics are to match them with certain plant groups. Perfect. In many cases, you don't know, but in many other cases, you do know. So this is how we reconstruct the environment. We use these little spores, and we use our knowledge to make connections to the parent plants. And thereby, we now have to analyze the pollen and the spores to reconstruct this. The cool thing about pollen and spores is they don't just occur in terrestrial settings, but they also, through rivers and the things, get washed into the sea. So we can also reconstruct environments neighboring to aquatic environments. So when you find a fossil that was deposited in water, we can still reconstruct what the world looked like. And we can do another thing. We can tell time very often. In this case here, we have a plesiosaurian. And the geologists who found it thought it might be from the Triassic, this oldest time of the Mesozoic. And they even named it Reticosaurus after the last time interval of the Triassic. But they were not entirely sure. And the problem is that plesiosaurians should occur only in what we know as the Jurassic. This is how they look in a reconstruction, but they were believed to only occur after this mass extinction interval that happens here at the Triassic-Jurassic boundary. So how can we tell how old this thing actually is? Well, again, we can look at the pollen and the spores, and we use them to tell time. And here is how it works. You see this beautiful section, and the colors are really kind of telling. So let me use another metaphor, which you might have seen last talk already as well. The cake metaphor is very common in paleontology, and if you imagine these little layers that you see here in the cake, imagine that over this section, over this cake, every time we take a little sample and put it in a bag. And now we're going to analyze each of these samples in the lab. And how do we do this? Well, how do I come from the rock part to looking at this spore that I showed you earlier? We need to get rid of the rock to do that. And we do that using HF, hydrofluoric acid. This is very toxic. Please don't try this. But if you think, I can solve some bodies away, no, this is not very useful because the hydrofluoric acid only solves silicates away, not organics. So if you want to dispose of a body, this is not your chemical to go to. The residue that we are left with, this is now what we can look at under the microscope. So we take a little bit of this fluid, we put it onto a microscope slide, and now we're going to look at it in detail. This is how it looks when you look at it with a microscope. And at this point, you might think, okay, what is she seeing? Well, let me use another metaphor. Since I come from Bonn, these are some very famous treats actually coming from Bonn. And imagine this is our sample, our residue, and in there, all the gummy bears are our pollen and our spores. What are we going to do with them? Well, we have them here in our sample completely mixed in. Now they're in the bowl, just like there. What we have to do now is that we have to count and match them to certain groups. Then we know how much we have of what. So if we have a lot of something, then we might assume there's a lot of that plant. Sometimes you might reduce it a little bit because you know that plant produces more than some others. But basically, the amount of spores and pollen kind of tell you what kind of plants occurred and how abundant they were. Now you look at your sample again and you count how much of what. Sample number 1. Here we see all these yellow and these green gummy bears, so they must be coming from this kind of area of the cake. The yellow ones seem to occur more abundantly throughout. The green ones seem to be quite unique to this layer. Next sample. We have yellow, red, and orange. And if we look at the cake, the yellow ones continuously occur, the orange ones as well. But the red ones, they are unique. They are actually time tellers. They are index fossils, and they tell us about a certain point in time. Now back to the real thing. These kind of red gummy bears, there are pollen that are so typical for certain times that we can tell that the Jurassic-Triassic boundary must be here. I told you already this is a mass extinction event. Now your next question is probably, how did the plants cope? Well, little excursion into the past. Here is the extinction interval. This is where all the other animals go extinct, but plants thrive. They still occur. And here are our real gummy bears. This is how they look. And in this zone, there are some that are really time-telling as well, and that assures us that this plesiosaurian is in fact Triassic, and that makes it the oldest plesiosaurian in the world. If we go into the extinction interval, we see a lot of these spores that we see here, and you see a lot of new taxa coming in this interval. So plants seem to be disturbed, but they go on, they survive, they have not as much problems as the animals during this extinction interval. And that is a general pattern that we see. Plants can't run away, so they have to have developed things that make them more resilient over time when they're stressed, compared to animals that seem to go extinct much more likely at those events. After the extinction, we have recovery phase and we have some new time tellers. All good. Now you have learned how we use plant fossils to tell time, but also you already saw there are different abundances. This is how we then reconstruct the vegetation. We can track the vegetation changing over time. So far, so good. Now, let us use this to a more exciting story, coming over to the angiosperms, the youngest of all land plants. Because while in the Mesozoic, in the old times, there were no angiosperms. When and where they actually occurred is one of the big questions in biology. So tackle it. We already heard it's an abominable mystery. Darwin was already busy with this publication, and as you see here, we have been moving from the Triassic through the Jurassic, and we are now in the Cretaceous. No one really knows when flowering plants actually occurred, but somewhere in this time interval, a revolution happened, because Earth's ecosystem significantly changed, because before there were gymnosperms and ferns dominating vegetation, but now a new player comes to the table and changes everything. But when and where? That's the question. Darwin looked at a publication here, which is actually a bit younger from up here, and he looked at that and he was really confused because looking at these fossils being presented, they look pretty much like old things he knew from everyday life around him. How could it be that out of a sudden so many diverse fossils occurred that look so similar to today? Because as soon as you moved a bit down in the fossil record in the Cretaceous, there was nothing. And he called that, this rapid diversification of angiosperms, an abominable mystery. If I paraphrase this, you can think of the dinosaurs going to bed. No angiosperms, no flowering plants. The world is entirely green. The next day, dinosaurs get up and the world is full of flowers and colors that were not there before. Of course, this is an exaggeration, but the fossil record looks a little bit like this because there is a huge fossil gap. First, you have very few fossils, and then out of a sudden, you have a lot of them in a huge diversity. How can that be? And this is exactly what he called an abominable mystery. Here, we can look at a graph not of plants, but of the bugs that are associated with plants. And it's a very interesting study highlighting again that it's not always about the plants, but about the impact that plants have on the rest of the world. And we see the bug families occurring through time. And you see here's a little peak in the late Jurassic, early Cretaceous, and then they're declining. Opposite direction, the angiosperms are increasing strongly, abundantly. Why so sudden? In between, there is the angiosperm radiation and also a fossil gap, because we don't really see this growing abundance. That's the suddenness that Darwin meant. And there are kind of two explanations for this. Either angiosperms did radiate super, super fast, and that's why we have this fossil gap from very little to a lot. Alternative explanation that we might have to think about it, maybe we don't see them, or we're not looking in the right way to find them. How can we change this? Well, this is what I'm trying to explore. What is the origin of angiosperms? Maybe we have to find a new way of looking, because scientists have been studying this question for more than 130 years, and we still don't know, and that's why it's still an abominable mystery. How can we solve this? Well, we learned a lot about plant microfossils already. And my big question is, how did the world change from this gymnosperm and fern-dominated world into something that is clearly angiosperm-dominated? There are different positions to tackle this. Amongst biologists, a lot of people are doing molecular clock studies. You heard already about phylogenies and all of those things. Well, you can approach things from a molecular point of view and calculate divergence times over time. And using them, you sort of predict at what time a certain plant group has to have occurred. And those studies claim that angiosperms pre-originated in the Jurassic, the time where the plesiosaurians normally should have occurred. But maybe they already occurred in the Triassic, just like the plesiosaurian occurring earlier as well than thought. Well, we don't really know, but this is a very old age. And the problem is that if we look at fossils, the world looks very different. That is that fossil gap, even dragged further down now, because some studies suggest to us we should be finding them, but we're not. So if we look at the fossil record, this is about the age that we can discern at the moment. And these molecular clock studies suggest that based on these genetic mutations that accumulate over time at predictable rates, give us a different age that we see in the fossil record. But now there is one little problem. These studies, they're usually using fossils as a calibration because there needs to be sort of a minimum and a maximum age for the occurrences. So fossils are not irrelevant. These molecular clock studies can't replace this. Finding fossils is still important. But the search for fossils has been declining because maybe people have not been finding what they wanted. But these fossils are super important because they function as an anchor for these other studies. So I think we should be still looking. And one person who has driven these things further through technical innovation is Else Marie Fries. If you have not read this very beautiful article in the New Yorker, I can very much recommend this. It's a beautiful sketch of a scientist's life, but also of a revolutionizing woman. And she has thought very much along the lines that I've just presented. Maybe we are not looking right. Maybe we need to find another way of looking at the fossil record. Rather than looking at macrofossils, Else Marie Fries has sieved the soil rather than looking for the things that you see with the naked eye with a magnifying glass. And what she found are the things you see here. Really, really small, but you can still see them with a magnifying glass. Bigger than pollen. Some of them even produce anthers with pollen in them, so we can now finally match some of the occurrences with parent plants. So this is a very important link, and these studies have pushed down the age and the diversification very, very much. It's basically thanks to Else Marie Fries and her co-workers who have illustrated this angiosperm radiation beautifully. But now my question is, we need to go further back, because if these molecular clock studies suggest that angiosperms occurred much earlier, how can we do this? And I think now we need to do the next step. If mesofossils already illustrated so much more that we can see, maybe microfossils, one level smaller, can help us even more. But the inherent problem is they are very small, so we need to find the right way for looking for them. I will not be as ambitious to start looking for them in the Jurassic, but let me show you a new method first, and if it is working here, then it might help us move further and further down as it works. So let us fix again. Fossil pollen has two huge advantages. It is very abundant and it preserves very well. And hunting fossils is a little bit like finding treasure for me. That's why there is the treasure chest in the back. And Jenny McElwain in the last presentation said, you will meet a lot of those paleobotanists who will try to find you the newest and the oldest. I'm exactly one of them. Let's go hunting for angiosperm fossils because we are met at an impasse. For many, many years, there was very little novelty, and let's try some new ways of looking to find some more things. These are all the records that we currently know for fossil pollen studies, and they're mapped along the world. And you see already that a lot of the fossils occur especially at mid-latitudes. The further down they occur here, the older they are. And you see that we have more in the mid-latitudes than much older occurrences here in the higher latitudes. But there is one off. This is the one that you see here in Portugal. And that's really strange. So I thought maybe look at that again. And also it's a nice place to study pollen. You can do beautiful fieldwork in this lovely village in Portugal. And actually this has been studied in times of Darwin already. So it is a very famous site anyway. And we have some outcrops along the coast. Everything that's green here in the nose of Portugal has these beautiful outcrops that you see here along the sea. As we're looking for fossils now, we have two main hurdles. Number one is if we're looking for something that's very old, well, we need to make sure that the age is what we presume it is. So making the age sure is difficult. The studies that have been done before, Torres Vedras and Cateficia that you see here, have been studied by Else Marie and co-workers a lot. But they have one limitation. The age is not so refined, and that has to do with being so much land inward. The good stratigraphic markers are usually found much better closer to the sea. Therefore, we decided that we're going to look at something where it's blue, indicating the sea, marine settings. And you see here in red star, that's our study area. And in the Hauterivian, we have a sea level high stand. Then the sea is retrieving a little bit in the late Barremian. And as we move forward in time, the sea is coming back. So we are always situated in this nearshore marine setting. And now the question is why? Well, there is one thing you don't find anywhere else on land, and that is bivalves. These kind of bivalves, they are growing like growth rings, and you can measure the strontium isotopes that are built in those growth rings. And those strontium isotopes are basically the superpowered carbon isotopes that you might know for carbon dating in much younger sediments. So as we are older, we need some more powerful things, and that's strontium isotopes, for example. This is what we did here. So for all the sample bags we also took bivalves. So we can measure the pollen, but we can also have something that gives us a more absolute age number. And this is something new that has not been done like that before. But every magic comes at a price. And this price I have to pay is, since we are in an aquatic setting, I find a lot of other things besides the ones I'm looking for. A lot of algae and things that live in seawater. So the terrestrial things get washed in, those are the ones I'm looking for, but I have a lot of admixture. And I have to delve through all of this before I find the thing I'm looking for. Okay, every magic comes at a price. Second hurdle. If we're looking for angiosperm evidence, we need to make sure that we're looking at angiosperms. And I told you earlier on that we are not for everything sure that they are angiosperms. And amongst gymnosperms, the seed plants that are not yet flowering plants, and angiosperms, there are some false friends. If you look at these two pollen types, they look quite similar superficially. They're not if you look inside them, but we can't always look inside them. So how do we circumvent that problem? Well, one way of doing it, if you want to be absolutely sure and convince all the devil's advocates, you can try to focus on this type of pollen. You see that it has 3 slits, one here, one there, and one there in this icon. And this is typical for a plant group that is called the eudicots. They are derived flowering plants. And their superpower is that they all have this type of pollen, and that makes them unique. If we find this, we can be pretty sure it must be a eudicot. So no discussion about potential confusion here with something else. So while for sure these must occur earlier, I now first focus on these derived groups, because then I have angiosperms for sure, and then we can start looking at the other ones. What has been found before? There is a single pollen grain that has been found before. And for 35 years, this has been the oldest tricolpate pollen, the oldest evidence for angiosperms. And it's a single pollen grain found from the UK by Norman Hughes, who was a professor in Cambridge. And he has done basically nothing else his entire life but sitting at the SEM and screening through samples and looking for angiosperms. And in all his lifetime, which was several years of work, he found a single thing that is still today, or until recently, the oldest evidence for eudicots. But now the question is, can we do better? And especially, can we date it better? Because this thing was not so bad as some other resources, but still there was some ambiguity about the age. So we try to improve the number of fossils and we try to improve the age. And the samples that I was working for or with at the beginning were standardly studied with this light microscopy. The problem was that they didn't find much, as you can see here. This is the interval we're interested, which would make things older, but here there's only some few not very spectacular things and no eudicots yet. But they were looking with light. And that's the limitation. This has been done for easily 100 years, looking with light, but maybe that's the problem. Looking for these angiosperms, they are tiny, they're really, really small. And maybe this is more like looking for a needle in the haystack. And the standard way of looking might not be good enough. And at the beginning of my research, I felt like this, like I'm not going to find anything. But there's some magic, fluorescence, as you can already see flaring up here. And if you switch on your fluorescence on a microscope that otherwise just uses standard light, you can see something coming up here. And as we switch on full fluorescence, you see it bright there. And that's an angiosperm. And that's the magic trick I use now for all the samples. I didn't use the standard bright field microscopy that everyone has been using forever, but we have been using fluorescence to find these nicely glowing angiosperms. Here you see the comparison, how much the difference is to find the thing you want. And if you do this, we see so much more. This is the improvement we can do with fluorescence. And not only do we find more fossils, we can also image them nicer, as you see here, if we use even improved microscopy techniques that are based on fluorescence. And here you see how nicely some of those pollen look like, and we can even look inside of them and not just on the surface impression, which looks a little bit like an SEM image, but it's not. It's only with fluorescence. And finally, the day that I found this, I actually opened a bottle of champagne because I was looking for this forever. We did find an angiosperm in those samples. We did find a eudicot angiosperm, a tricarpate pollen. This one was for sure one. The other ones might be, but this one for sure is an angiosperm. And you see clearly those 3 slits here, and you even see this beautiful surface ornamentation, which looks like little walls on top of there. We see a lot of detail, which was not visible before. And you see here in the light, a lot of the details are obscured. So new microscopy techniques definitely help to image this. We didn't only find this one, but we found more champagne. Lovely. So we find this one, and this one, and this one, and also some of them, you actually have to move them to even see the 3 slits, and that might have prevented people from recognizing them before. Also because here in these, the slits are not very well visible. So one of the problems might have been we have overlooked them because they were too small. I actually only found them with fluorescence, and some of them are so obscured because of a bad angle that you simply overlook the important feature. Now, all of them are from the same sample, about this age, 122 million years, so that's already pretty good, knowing that we were not so sure, but they are still from the late Barremian, which is probably similar to the previous oldest grain. So I've been looking further and hoping we find some more, and we did, of course. So this one here was recently published as the now oldest angiosperm evidence. And you see here from the scale on the right, it's the lower of the 2 grains, so this is significantly older, about 1 million years older than the other one. And considering that the Barremian as such is only 4 to 5 million years long, 1 million years in this time frame, that's actually a lot of time for evolution. So this is really, really nice and interesting, and it has shown that the way you look changes very much how much you can find. So we definitely improved on this one pollen grain 35 years later. Phylogenetic studies should not cite this one anymore, but actually we have a better one now, an older one, and we could push the age of angiosperms down using these very intricate microscopy techniques and looking at these super, super tiny fossils. So what we did, I showed you here at the beginning, it was so odd that this thing was so high up there. Actually, we now pushed it significantly further down, and now it looks much more similar to the rest. And if you look all of this together, people still think that angiosperms originated probably in those mid-latitudes. But for me, it's not so much simply about pushing the age down for this one record about 2 million years. This is about the principle. If we can push this age that much down, what happens to all the other records if we look at them in the same way? No one has done that. Many of them have been studied 30, 40 years ago. Now, I think there's a huge need to reevaluate these things. And that brings me to my summary. You have learned so much about pollen and what they can do for us studying the past. And for sure, you have all learned that the sporopollenin is the mithril of botany, this enclosing, very beautifully ornamented structure surrounding the pollen and the spore wall, which is so durable that we can study it over millions and millions of years. We can also use these pollen to date things, especially in times of extinction. Sometimes these plant microfossils are the only thing left, nothing else, no macrofossils, no plant microfossils, nothing else. But in other times, we use that pollen occur in terrestrial as well as aquatic settings. And then we can use other fossils to give a precise dating, and we use the co-occurrence on land and water to correlate things over large distances. Then, of course, this is probably one of the cool results from this study, that we could push down the age of angiosperms 2 million years further down. They are now occurring at about 123 million years ago, and we have much better dating than all the things before. We have more fossils than there were before. So all of this is also helping producing that anchor for all the phylogenetic studies, those molecular clock studies, and hopefully this brings us a bit closer to solving this abominable mystery and to push the age of angiosperms further down. But those of you who think, ah, what are 2 million years in the time frame of it all? Well, perfectly fine. I think this, for me, is much more about the principle, what this technical innovation can do for you, and that it is very much worth looking for more fossils because I think there is a promising future to find more. And who knows, maybe we can push those ages much further down and come closer to those predictions that come from molecular studies, maybe suggesting Jurassic or even a Triassic age. With this, I can only thank all my many colleagues and friends who have been working on this with me or supporting me along the way, all the funding agencies who have paid for this to show you all these beautiful images. And of course, for the invitation to share my passion for these huge, small microfossils. They are huge, I think, although they're so small. And I thank you all so much for sharing this time with me and listening attentively. Thank you.
Thank you so much, Julia, for a fascinating talk, and wonderful to come to the end with such a great talk on the origin of angiosperms as deduced from the pollen record. Can I just kick off with the first question? You've used the tricolpate eudicot pollen as a sort of proxy for your aging of the origin of angiosperms, but what can the monocolpate pollen that you see in the fossil record tell us about the really early stuff that involves the what we call today the basal angiosperms and the magnoliids, and also the relationship between magnoliids and monocots and their relationship with particularly Chloranthaceae. There's a few questions in there, but it's really about what the monocolpates can say about this.
Yeah. As I said, the starting point for our analysis was working on the tricolpates because there no devil's advocate can claim that this is not an angiosperm because they must be. But as you saw already, these monocolpates or monosulcates, so those with one slit instead of the three, they are very, very telling. And I think the next goal is to push those ages, the blue lines that you saw below the tricolpates, further down, because I think that's the really interesting part. And some of my students are working on that at the moment so that we can hopefully push down the age significantly further. And the thing is, with the new methods that we use, we see much more detail that we could see before. So the problem with the monocolpate or monosulcate is that we don't have much features if we look at them with light. But now that we have more possibilities to image them, we might discern more features that might be typical for certain groups that we didn't see before. And we can, of course, use the excellent records of plants that we have from gardens and so on to have a reference collection to match those features with them. And you can see a lot more things with the fluorescence that you could before.
And what about the origin of the monocots? Can anything be inferred about that from the fossil record and their relationship with the rest of the monocolpates?
At the moment, I'm really not sure because, of course, distinguishing between a colpate and sulcate is a problem as such already if you look at them just from the surface, but also making those additional inferences for certain groups, I'm not confident to make any claims on that at the moment, but who knows? We can find looking at them in more detail.
Okay, thank you. Now let's go to the Q&A from the audience. So the first question is really about what microfossils can tell us about when lichens first appear. This may be stretching it a bit for your talk, but can you say anything about that in terms of microfossils?
Well, it depends, of course. A lichen is a symbiotic organism.
Mm.
So, what remains are we actually looking for? Now, I'm not looking at those earliest fossils. For that, we would have to go into the Paleozoic, and I don't know enough about it. But in theory, the spores from fungi, some of which are very typical for certain lichen, they can preserve very well. Actually, there's lots of people studying fungi. And some of the structures that lichen produce can fossilize as well. So we don't just find spores and pollen, we find all kinds of admixtures of plant remains that are organic in our samples. So you saw those clusters, you saw pollen spores, algae, but also wood and cuticle. There's all kinds of things in there. So if you find remains that might match the morphology of a lichen, you might recognize it. But I can tell you from another little thing, I'm working on elatirs, which are actually from hornwoods and a lot of mercantia who occur very early in time as well. Palynologists simply might not have recognized them as what they are because a lot of the palynologists are geologists, not biologists.
Mm.
So if you have that biological morphology knowledge, you will recognize them. But most people who have been studying it haven't. And I think that's another problem for the angiosperms. A lot of the people who have been working on those older records, they have been geologists. They were looking for index taxa. They were not looking about the biology or the shape changes that might indicate anything about their associations with the plant, the parent plant. So in short, I cannot really tell you about any details on the lichen, but it is possible to find remains in the samples, but that's not my specialty.
No,
For that,
well,
you can try go to Paul Strother or Charlie Wellman. If you have something in
mm,
particular that triggers that question, they would know.
yes, good couple of names there to conjure with. Um, thank you. And going back to palynology and particularly the fluorescence work that you're doing, um, have other people been using fluorescence on to look at the spores of earlier um, land plants?
Yes. So it actually was Mr. van Geisel who started fluorescence work in the 1950s already. And he started to do it to look at the whole assemblage. Cuticles fluoresce very beautifully as well. And he originally wanted to discern between marine or aquatic samples from terrestrial because algae have a stronger fluorescence. So if you have amorphous matter, it glows a lot and you can tell immediately, is this aquatic or marine versus terrestrial? And that, of course, has started people looking at pollen assemblages with fluorescence, also earlier records. But I think it has been forgotten for a while. But I think in the last 5 years, also a lot of macro people have been using it to find the spores and the pollen on their macro remains, as I showed you earlier on with the flower and the bumblebee. So yes, people have been using fluorescence to image things even from the Rhynie chert, some very early land
Hmm,
plants. You can use fluorescence sometimes depending on your preservation, and people are doing that more now. So yes, they have been looking at it with it.
thank you. Now, another question. Is it possible for molecular biologists to get DNA out of the pollen samples that you study so that you can use your microfossil data to gain insights into molecular clock hypotheses?
That is the wet dream of every biologist, of course. The quick answer is not really. People are working on ancient DNA, but ancient is not really ancient in the sense of working on the Mesozoic. So if you use some, what I would call a subfossil, so going back into the Cenozoic, people are working on that now, and there are some more promising studies on that, but these things are super young in comparison. Basically, it's yesterday in compared to the things that I just showed you. So the quick answer is not at the moment, but I wouldn't exclude it in the future. Who knows?
Who knows, indeed, yes. And then another question from a regular of ours who comments, fantastic talk, thank you to begin with. As a non botanist, please could you explain the evolutionary advantage of tricolpate pollen, or indeed why there are so many beautiful shapes? Two super questions there in one.
Yes. And I think this goes more in almost into the philosophy of biology. There is this analogy of, that in churches you have those corners that are produced by the architecture to keep the church stable. And as a result, artists have been starting to do a whole own art of coloring exactly those parts of the church.
Mm.
Now, they were produced because those things were there. Just because something is there as a morphology doesn't need to mean it has a function. It can mean that it has no bad function, no disadvantage. So it doesn't get selected away. So just because we see something doesn't mean it has to have an advantage. It can, though. So some patterns that we definitely see is that, for example, in lycophytes, something occurs that the male gametophyte, which is sort of one generation of a plant, it actually lives in the spore for a long time. And those spores, they are super different on both sides, which basically means it's like your jam bread. It always falls on this side. So sometimes the ornamentation can have the function that it always sits in the right way. That's one possibility. And in pollen, we see that the ornamentation follows general principles. If it is wind dispersed, it is small and has no ornamentation. It is animal dispersed, especially from bees and so on, it's sort of a middle size range with nice sticky ornamentation. If you have animals pollinating, like bats, for example, they have super huge pollen that stick nicely around the nose of the bat. So yes, there are definitely advantages in the ornamentation, but not always and not necessarily.
Okay, but in terms of the three
Yeah,
the
sorry.
three
The three,
three three exits for the pollen tube through this very tough surface, do you
the
think it gives the pollen tube more opportunities to escape from its really tough case by having three rather than one?
There
Uh,
have been people arguing that, but the devil's advocate argument would be, what do all those pollen do that have no opening at all? And a lot of the early angiosperms
mm,
actually have that. So yes, it's probably likely that those three is sort of a good number to work. And as you see, if you sit on the stigma, to have a good exit route to grow your pollen tube, and angiosperms are really good at growing pollen tubes really fast. This might have been even improving on that. And maybe that is indeed the reason why the very early angiosperms are not so fast and take everything a bit slower than the others. But well, again, there is no definite answer to this. This is a theory. Some people go for it, others don't.
okay, I'm just looking back. We've got another question that's just come in. Um, now, this is a good one. Do you think Cretaceous dinosaurs may have suffered from pollen allergies, which of
I
course
thought that one
which is of
before.
course it raises the question of when an allergy to pollen arose originally in whatever animal developed it.
I would actually say that the dinosaurs would probably be happy about all those angiosperms who are pollinated more by insects, which usually means that they produce a lot less pollen. I would rather assume that if dinosaurs were allergic, in fact, they might have been much more likely allergic to gymnosperms, which
Yes,
produce way, way bigger loads of pollen and already way earlier than the Cretaceous, because one group of gymnosperms that actually thrived during the times the dinosaurs reigned the world is an extinct group nowadays, and they make a pollen that's called classopolis. It's like three balls that sit nicely together. And no matter in which Mesozoic sample you look like, they are everywhere. So if I had to take a bet that dinosaurs might have been allergic to anything, that would be it.
okay. Right, let me just check. No, there are no more questions in the Q&A. So I think we'll call a halt with perfect timing from you, Julia, in our final lecture. Thank you so much, and a big virtual round of applause from everyone in the audience. And thank you all for attending these lectures. And I'll just say, please join us in person if you can for the winter lectures that begin in January. These are not science lectures, but they are lectures that relate to plants in all sorts of contexts, including gardens, garden design, garden history, etc. So for now, thank you, Julia. That was a lovely talk and great to end with angiosperms and pollen, and wish you very well in your research and look forward to meeting you in Bonn in due course. So thank you and good night.